Good morning, children. Yesterday I explained you about the difference between the osmosis and diffusion, and today I'll tell you about the endosmosis and exosmosis. See, osmosis is studied under the two headings. First is endosmosis and second is exosmosis. Endosmosis means entry. Okay, endo means entry. So endo means movement of water molecule inside the cell. ठीक है, endo means entry. So what does it mean? Movement of water molecule inside the cell. So suppose we place a red blood cell in a beaker. Suppose we place a red blood cell in a beaker. This is RBC. Okay, so suppose you place a RBC in a beaker filled with water. This is water. So what will happen? The concentration of water inside the RBC is less, but the concentration of water outside the cell is more. ठीक है ना? So due to the process of osmosis, water will flow inside the RBC. ठीक है? Water will flow inside the RBC and RBC will get swell up. So this is known as a endosmosis. Clear? So you can define in this way endosmosis. It is the movement of the water inside the cell when the cell is placed in a hypotonic solution. And hypotonic means a solution whose concentration is less than that of the cell sap. Cell sap here, the cell sap is RBC. Next is exosmosis. Same way, in beaker, if we place the RBC in a hypertonic solution, this is a hypotonic solution, and here this is a hypertonic solution. Hypertonic means a solution containing less water and more sugar or salt or anything. Okay, so in this case, यहाँ पे RBC, this is RBC and this is salt or sugar or anything, whatever. ठीक है ना? So in this case, RBC will lose water by the process of osmosis. So that is known as exosmosis. ये क्या करेगा? लॉस कर देगा water. And that is known as exosmosis. So, how can you define exosmosis? Exosmosis is the movement of water outside the cells when a cell placed in a hypotonic solution. And hypotonic solution कौन सा होता है? Hypotonic solution whose concentration is more than that of the cell sap. Here also the cell sap is this. This is RBC. And the cell become flaccid by the movement of water outside. Okay. Next topic is three types of solutions. See, three types of solutions कौन कौन से होते हैं? Hypotonic, hypertonic and isotonic. अब as I already explained you about the little bit about the hypotonic and hypertonic while explaining the endosmosis and exosmosis. So when we study the osmosis, there are generally three terms, a very important term, hypotonic, hypertonic and isotonic, and this topic is very important topic, and one question will definitely come in exams. Okay, got it. So suppose we have three beakers, 1, 2 and 3. Suppose we have three beakers and first beaker contain hypotonic solution. Hypotonic, a dilute solution is always a hypotonic solution. आपको पता है, मैंने बताया था dilute solution क्या होता है? Hypotonic solution. A solution with more water and less solute is known as a hypotonic solution, and hypertonic solution is a concentrated solution. Hypertonic solution क्या होता है? Concentrated solution होता है which is less amount of water and more amount of solute. And isotonic solution is a solution which has both present in equal amount. ठीक है? Both present in equal amount solute as well as the solvent. So this is the difference between this. So hypotonic solution is always water. Hypotonic solution always sugar solution or saline solution. And isotonic equal amount of solute as well as solvent. Okay, and even when you place any cell, so no change in the cell when placed in an isotonic solution. Now the question arises, what will happen in a cell when placed in a hypotonic solution? अगर hypotonic solution में कोई cell लग दोगे तो क्या होगा? So when cell is placed in a hypotonic solution, the cell will have less water molecule as compared to the surrounding. ठीक है ना? Cell has less water molecule. यहाँ पे क्या? This is water. It's obvious. ज़्यादा molecules हैं. So what will happen? The cell will have less water molecule as compared to the surrounding. The water is going to enter inside. Water will go and water will enter inside the cell. A cell will always swell up when placed in a hypotonic solution. And in hypotonic solution, you know that maybe always a sugar solution or a brine solution. So once you place the cell, what will happen? The cell will always get shrink. So, and when placed in an isotonic solution, there will be no change. So these types of solutions, three types of solution: hypotonic, hypertonic, and isotonic. Next term is plasmolysis and deplasmolysis. Plasmolysis means shrinkage, or you can say flaccid or shrinkage. And deplasmolysis is also known as turgid cell. So plasmolysis is a condition in which when you place a cell in a hypotonic solution, cell will lose water. Means whatever the cell you are placing in that solution, that has to have more concentration as compared to solution which is taking. Clear? So in the beaker, suppose we have sugar syrup. Okay, in the beaker, suppose we have a sugar syrup. Now this cell has more water content as compared to the sugar solution. This is a sugar solution and this is cell, and this cell has a more water content as compared to the sugar solution. So when the cell is placed according to osmosis, cell will lose water and shrink or flaccid, or cell is plasmolized. ठीक है, क्या करेगा? Shrink हो जाएगा और किस में convert हो जाएगा? The cell will get plasmolized. Now if you place this plasmolized cell, the shrink cell, in tap water again, this cell is going to swell up. Okay? So that is known as deplasmolysis. But if you take this cell and place in water, so this is a turgid condition and will not be called as deplasmolysis. Clear? So you can say deplasmolysis is always a reversal of plasmolysis. So deplasmolysis is always a reversal of plasmolysis. So when cell is placed in tap water and it regain its shape, is known as deplasmolysis, or you can say reversal of plasmolysis. So this is all for today. Thank you.